You're live. Okay, it says that we are live. Perfect. Hello, everyone. Welcome to our session. I'm very excited to introduce our first speaker. We have Jing Cai, who's from Mass General Hospital, and she's a postdoc also at the at Harvard Medical School. And she will tell us a little bit about her work on human language at single neuron level, and we're excited to hear from her. Take it away. And please, everyone, remember to put your questions in the Slido, and we look forward to it. Go ahead.
Hello, good afternoon. It is my great pleasure to present my current work at Professor Ziv Williams' lab at the single neural activities during human language comprehension. Okay, so human languages are extraordinarily rich, and it can convey a lot of detailed information as well. So in the figure that I'm showing, these two are examples that are showing how rich and amazing human languages can do. And in a sentence, usually in English, that a sentence is composed of words, and we have more than 100,000 words in English. And the words has to be organized in a certain order for the listeners to understand, making the language very complex. And human speeches are also highly dynamical in the sense that it's usually carried at a speed of 150 words per minute. But our brain, or the listener's brain, has no trouble in processing all of this information, which is quite a computational feat. Okay, so there are quite a few of the interconnected brain regions that are underlying the human language comprehension, or specifically the semantic processing. And by semantic, it means the meaning of words and sentences. So the figure on the left are showing the previous study of the fMRI images, and these are showing the regions that are specifically significantly contrast in the semantic processing. So as we can see that there are quite a few of these brain regions, including the temporal lobe, the parietal lobe, and as well as the part of the prefrontal cortex. Although quite a lot of work has been done previously using the imaging measurements like fMRI to study the language comprehension, but little work has been done to study the single neural representation during human language comprehension. So note that the single neural recording has a very high temporal and spatial resolution, which are very good for these high-level cognitive processes. So the question that we are specifically interested in is how are sentences represented at a single neural level during language comprehension? So we utilize a very unique opportunity to record single neural activities during craniotomy. So we asked the participants to hear naturalistic sentences, and I'm showing one example in here. In the meantime, we record from the frontal cortex about the voltage of this brain activity. And from the voltage, we can isolate a single unit, and then so we can get the single neuron activities from here. Then we can also decompose a sentence into a couple of linguistic aspects. So here I'm showing 5 of the examples of these aspects that we examined. So the phonemes are encoding the pronunciation information of a word. So this is actually part of the auditory information. And then for the word meaning information, we categorize words into a couple of groups, and each group represents a unique part of their meaning, such as whether it's a name or action or family, and etc. And then we also study the compositional meaning, and this is more like the semantic role in the sentence of a word. So further, we have the single word syntax, whether it's a noun or adjective. And then for working memory part of information, we use the word position, which is linearly increases with the number of words. So having all these aspects in a sentence, the question now we ask is, do single neurons encode any of these aspects directly or not? So let me show you 2 examples from single neural activities. So the cell on the left are the neurons that is specifically tuned to select one of the word meaning clusters, which is the action. So what it means is that whenever the participants hear words like chased, run, walked, reached, then this neuron is going to show a higher firing rate compared to the other words that the participants hears. So I'm showing another example on the right, which is the neuron that carries the grammar information, where specifically these neurons are responding to the adjectives. So having recorded hundreds of neurons, now we can do some statistics in asking what are the ratios that these neurons are specifically encoded to each of the aspects of the language. So the results is listed on the below. So on the phoneme part of information, we found that they are less than or equal to chance level of the percentage of the neurons that are encoding the phoneme. So this means that these neurons are not specifically tuned to encode the auditory information. However, when we are examining all the other aspects, we found that there are significantly higher number of neurons that are tuned to all this spectrum of information, ranging from single word meaning, grammar, compositional meaning, and the word position. So all of these together are suggesting that the single neurons are directly encoding a variety of the linguistic aspects. So now we know what each single neuron are doing. And these single neurons are like each pieces of a puzzle, whereas the whole puzzle, when you put them together, will form the network that is underlying the human language comprehension. So the question now is, how are these neurons assembled together? And specifically, what are their collective behaviors of this neural population? So note that this is quite a complicated process in the sense that not only there are lots of words in English, but also the order of the words matters to infer the sentence meaning. So in here, we turn into the deep learning models and natural language processing. So with the rapidly development of recent advancement in deep learning models, there are quite a lot of applications for these models. So for example, the Google Home can do quite a good speech recognition and question answering. And for Google Translate, after incorporating the deep learning algorithm, it has a good advancement in the performance of translation. And recently, OpenAI just announced a new model called the GPT-3 model, which is a language generative model that can perform a high level of the processing and performance in a variety of language tasks. So we borrowed a pre-trained GPT-2 model, which is a previous version of the GPT-3 model. So this model is capable to not only encode the word level information, but it also has the whole high level compositional information being processed in the model. And as a result, this model can have a very good performance in predicting the upcoming words without knowing it. So for example, if the previous words is the baseball team is as the input to the model, then this model is able to predict that the next word is highly likely to be playing, which actually makes sense. So all of this language processing is done via 12 modules, and these modules are connected via a sequence way in here. In between two of these modules, information were carried via 768 of these artificial neurons. And now the question is, do we ever know anything about what is actually processing in this artificial model, or can we open this black box? So previous work has shown that this artificial model is processing more of the word level information in the lower layers, whereas in the high layer, higher up there, it is processing more of the integrated and highly dynamical information that is incorporating the compositional meaning. Okay, so now we have the brain neuron, and we also have the artificial neural activities. So the question we ask is, can we build a map to link between the brain neurons and the artificial neuron based on their activities? So we searched through all the pairs between these two kinds of neurons, and we ask whether they are significantly correlated in their activities or not. So then, after building this map, we can ask what are the percentage of the brain neurons that showed their synactivity paired artificial neurons. The result is showing on the right-hand side that the x-axis are the GPT-2 layers that the artificial neurons are obtained, and the y-axis are the percentage of the brain neurons that has the artificial pair existed. As we can see that there is an increasing trend of this percentage of brain neurons, and it reaches a plateau at around 25%. It means that from the recorded neurons that we have, a quarter of them actually specifically tuned to the artificial neurons that are obtained from the higher layers of the GPT-2 model. It also implies that the single neurons are assembled in the way that is best described by the top layers of the GPT-2 model. Further, we can combine these analyses together. So the x-axis are showing the probe analysis, and the y-axis are showing the single neural selectivity to the artificial neurons. So what we mean here is that each dot represents a brain neuron. And let me take an example. So on the second column, so we have 8 neurons on the top of these GPT-2 layers. So what it means is that there are 8 neurons that are specifically tuned not only for the word syntactical information, but also they have the counterpart of the GPT-2 neurons that is locating on the top layer of the GPT-2 model. As we can see from this image that the brain neurons are widely distributed across different aspects of probe or information of the language, as well as the GPT-2 layers. The more interesting part, or remarkably interesting part, is on the rightmost column. And these are the neurons that show the selectivity to the GPT-2 neurons, to the artificial neurons, but they do not show any selectivity to any of the probes or aspects of language information that we examined. And because we see an increasing trend with the GPT-2 layer, so it implies that these neurons are encoding very high-level compositional language information that is pretty much beyond the reach of all the traditional probes that we examined. Further, we want to ask, in the population level, how much variance can the artificial model explain? So to do that, we run some models with the artificial neurons as the input to predict the brain activities. And then we got the log likelihood as the pseudo R-squared, which is a representation of the variance explained by the artificial model. So the results is showing on the right that we see there is an increasing trend of the variance explained with the GPT-2 layers. And this is consistent with the single neural selectivity results from the artificial model. And this also indicates that the collective behavior of the neurons were best described by the higher layers of the artificial model. Further, because I have just shown that the artificial model has the property of to predict the upcoming words, even given the previous words. So now I want to ask whether the brain neurons has the same property or not. So there has been quite some debate over the hypothesis that human language comprehension requires future word prediction. And this hypothesis is remaining controversial for quite a long time. And the major reason why it's controversial is because there's a lack of experimental evidence to support for the prediction. So now we have the single neural recording from the single neuron level. I want to ask whether we can examine this hypothesis at this level. So here I use the current neural firing rate to predict the next word semantic category. The result is showing on the right that we found the decoding accuracy is locating much higher than all 1,000 iterations of the shuffled control. So this indicates that these brain neurons are indeed encoding the future word prediction during the language comprehension. It also implies that while you are now listening to my speech, your brain is constantly making predictions of what the word that I'm going to say. All right, so to sum up, we did single neural recordings during human language comprehension. We found that single neurons are specifically tuned to a variety of information, ranging from semantic, syntactical, and working memory. Then using an artificial network, we found that these neurons are also highly tuned to encode the compositional meaning information. And all these neurons together has the property to predict the future word. On the population level, we found that the population variance can be better explained by the higher layers of this artificial model, meaning their collective behavior is best described by these upper layers of the GPT-2 model. So this implies that these neurons may evolve collectively to encode this highly dynamical of the language comprehension. So thank you for your attention, and now I'm ready to take questions.
Thank you so much for your talk. There's a number of questions. I just want to appreciate how much work goes into that, but also mention that there are many models that can predict how something else is going to come up in the brain, and not just language models. So the first question is from Panghao Shantan. He's asking, do you have an example spike raster plot of a brain neuron that would be classified as behaving similarly to one of the GPT-2 neurons? And what is the criteria?
Oh, so I don't have an example that I'm showing in here, but the way that we are relating them is to so we have the activity, which is a one-dimensional time sequence. So we just take the direct correlation to them and then run a pretty much like a basic linear regression. And then I did a t-test with the null hypothesis of whether the beta or the coefficient is 0 or not. So the rejection, if they are showing highly correlated information, the meaning that their beta or the correlation are higher, and the rejection will be like the p-value. And I also did, because we have multiple comparison, so I also need to correct for that. Yes.
The second highest voted question is from Omri Barak, and they ask, is the prediction using both the history of words and neural activity? Does neural activity significantly improve prediction from words alone?
So we did not, okay, so to answer the data that I showed specifically did not add the history of the words. It's all purely using the neural activity to predict the future word semantic category. But we did do a control, which is adding the current word. So we didn't do all the history of the words, but we did add the current word semantic category as an independent variable to predict the next word. And then the control is to shuffle the neural activity while keeping the current word semantic category. Okay, if I'm clear enough. So this is, I guess, there are a couple of issues there are a couple of controls that we did. And we didn't, it's a little tricky to compare directly whether adding other dimensions in the features will have a higher performance or not, because it requires quite some details in the model itself, such as what regularization you choose and what control you choose. So but we did in adding the current word semantic category, and we found that there are still very high separation between the actual prediction and the control.
And if you have a quick answer so that we can switch to the next talk, how are you determining correspondence between biological and artificial neurons? And it would help if you also tell us how many neurons in the brain, how many words, how many concepts, words, sentences, etc. were you trying to match?
Oh, so we just directly use our hundreds of brain neurons that we recorded, and in the artificial network, each layer has 768 number of neurons. So we did a pairwise comparison with the correction of multiple comparison. Yeah, I think, sorry, I forgot the second part of the question.
Oh, that's totally fine. Thank you so much. You did a fantastic job of answering a lot of questions. And if you now unshare and mute yourself and turn off the camera, then Jonnathan, can you please turn on your camera? Jonnathan? Perfect. And you're muted. Could you unmute yourself also? And please share your slide. We only see the... You're still muted. Okay. So we're going to wait for Jonnathan for a minute.
Okay. Can you hear me all right?
Yes, please share your slide.
Sorry about that, guys. I am going to share my screen here. Sorry, can you see?
Yeah.
Okay.
So Jonnathan Singh Alvarado will talk to us a little bit about neural dynamics in basal ganglia underlying birdsong practice.
Okay, thank you so much. Sorry about the chaotic beginning here. My name is Jonnathan, and today I'm going to be talking to you a little bit about the work I've done in the last 6 years concerning neural dynamics in the basal ganglia when birds practice and perform. And really, in graduate school, the question I became interested in was how our brains regulate motor variability. And just to illustrate that a little bit better, I'd like to show you an example. So imagine your favorite musician. In this case, this is one of my favorite musicians. His name is Victor Uden. He's a really good bassist. And your favorite musician has probably played in a wide variety of venues. So when playing in a smaller, more intimate venue, for example, BU Cafe, which is a small dive bar downtown in the city I live in, Victor might play more loosely and allow himself to improvise or explore musical space. And we can visualize this with a plot like this. On the x-axis, we have the different potential, let's call them musical variations that Victor can perform, such as different notes, phrasing, tempo, key, that Wooten could use. And in the y-axis, we have the probability of him selecting each variation. Now, in BU Cafe, this might be a relatively wide distribution reflecting the higher variability as associated with the exploration of musical space. And this variability is not just random, it reflects a meaningful exploration that can be used to improve performances in the future, for example. But conversely, in a more high-stakes venue, like, for example, Carnegie Hall, the musical variations that he would use might be more constrained by the importance of the concert as he tries to deliver a more precise performance. And going back to our variability plot, this would be reflected as a sharper, more precise distribution of selected actions. And I just want to highlight that here, really successful behavior depends on the ability to switch between this high and low variability state, depending on what context you might find yourself in. So the way I tackled this in lab is using songbirds. And there's really two main reasons to use songbirds for this. One is that they exhibit two very distinct ways of singing. They can sing alone, and they'll just sing spontaneously thousands of times throughout the day. We call this undirected singing. But arguably in a more important fashion, they can sing towards a female songbird, a female zebrafinch in this case. And I want to highlight also that this transition from this undirected practice solo state to this directed, female-directed performance state is associated with a change from high variability to low variability, respectively. Now, just to familiarize a little bit more with how we quantify behavioral variability in song, this is a spectrogram of a zebrafinch's song. This is called a motif, and they just perform. This is the phrase that they learn and perform over and over. Zebrafinches only learn one type of motif. And to quantify this a little bit more easy, to quantify the spectrogram, we tend to divide it into discrete elements that we call syllables. So here that would be A, B, C, D, E. Now we can take an example syllable, for example, syllable E, and analyze this individually and ask, what is the pitch of the fundamental frequency? So that's just the lowest part of the syllable. And as the bird sings over and over again, we can get the pitch contour for each syllable rendition, plot it over itself, and use some simple metric of variability, like, for example, standard deviation, the coefficient of variance, some measure like this, to get an idea of how variable the spectral qualities of that syllable are over time. And I was telling you before that directed song is less variable than undirected song. So for a distribution of undirected song like this, for an example syllable, the directed distribution for that same day would look a little bit like this, where you can see it's more constrained, variability is reduced. And even though, you know, if I went and saw this spectrogram for the undirected singing and the directed singing, it doesn't look very different, but to the females, it's extremely salient, and they greatly prefer, for example, in this double speaker assay, they much prefer hanging out and spending time by the speaker that's playing the mate's directed songs, but in comparison to another speaker right in the same cage that's playing the mate's undirected song. So this seems to be a pretty ethologically relevant behavior that's readily perceived by arguably the most important listener, in this case, the female finch. The second reason that we study songbirds is because they've evolved a network of specialized brain regions that are only dedicated to the acquisition and the performance of song itself. So it makes it way easier for us to draw links between behavior and the underlying neural substrates. And within the system, my work is focused on the song specialized basal ganglia. So that's area X right here. And area X is critical for the bird to learn its song, but today I'll hopefully also convince you that it's important and plays a key role in the contextual control of motor variability. And just to orient you a little bit better to how the songbird basal ganglia is oriented, it shares a lot of similarities with its mammalian counterpart in terms of anatomy, physiology, and connectivity. It receives excitatory input from the premotor region, HVC. It outputs to the thalamus, and that forms a loop that eventually can influence motor output regions, and it directly influences the performance of song. And one really important difference, however, is that area X has both striatal and palatal components anatomically intermingled in one region. But the connectivity and how information flows is thought to be still pretty similar to mammalian systems. Now, having all this in mind, my goal was really to monitor the activity of area X, specifically spiny neurons in area X, because of their importance in striatal computations and the fact that they make up over 90% of area X's population. But this was really hard. SNs, or spiny neurons, are very difficult to record. They're very small and they're very sparse in their activity. So we decided to tackle this with calcium imaging. And the way we went about this was to combine calcium imaging with miniature microscopy so that the birds could behave freely. This is just a simple diagram explaining how we did this. First, we delivered GCamp to area X, specifically to spiny neurons with area X, using a promoter called CAMK2, which all you need to know for this talk is it delivers the GCamp specifically to spiny neurons within area X, and then we could monitor their calcium signals. To get optical access to this neurons, what we did was put in a Grin lens, and this was a little bit of a special Grin lens because it was attached to a prism at the end, and what that allowed us to do was avoid important real estate that was dorsal to area X that was also part of the song system downstream. And it also allowed us to minimize damage in the plane that we were imaging, because we were getting a reflection of 90 degrees that allows us to image orthogonally into the front of area X. Histologically, that looks something like this. You're seeing area X, and it's infected with GCamp in MSNs and spiny neurons only. If we were to look out the side of this prism, you would get a field of view that looks something like this. And you can see the blood vessels, you see the bright spots, those are cells. We can extract the spatial footprints of our neurons of interest from these videos and then analyze those. And this is what the bird looks like when it's actually wearing the microscope. I'm going to show you next a video of activity as the bird is performing, so singing undirected song. Hopefully you can hear the sound, but so this was really exciting for us. This was after a long period of troubleshooting and failure. This was after 2 years, I think, of really difficult troubleshooting. And from this activity, I can show you the example of 13 neurons from that field of view. Above, in the top, you're seeing spectrograms for the bird singing undirected song, marked by the blue bars. And then you're also seeing the activity of those 13 neurons aligned to that. And there's two things that pop out to you instantly from this. One is that activity is very related to the actual act of singing. And another one is that it is not exactly the same from rendition to rendition. It's quite variable from the rendition to rendition. So to better explain this a little bit, I can show you a representation like this, where this is a heat map of the activity of the neurons in a song, one of those songs. So on the y-axis, you have the list of neurons. On the x-axis, you have time. The white dashed line is the onset of a song. We can sort that activity, just get some continuous representation, just sorted by timing. And then we can ask, if we use that same sorting for the next song, if it is true that activity is pretty stable from rendition to rendition, we should get back a similar organization. But instead, what you see is that you get the addition of new neurons on top, and then you also get dropout and slight jitter in the neurons and the other neurons that were active in song 1. Now, I'm not going to go into the analysis of how we quantified all of this in the interest of time, but suffice to say that activity was very different from rendition to rendition, which was a little bit unexpected to us. And I told you before that we really wanted to compare this activity between directed and undirected singing, so I'll cut to the chase and just show you what that looks like with 2 videos. So this is undirected song. Just like you saw before. And now this is in the case of performing to a female.
Jonathan, can you unmute your screen as well? Your screen share is muted, I think.
I am not sure. That's unfortunate, but that's the last piece of sound. It's the same sound in both videos. One has activity and one doesn't. Sorry, I think I would need to minimize my presentation for that. I can't seem to
That's
do
okay.
that from
Please
here.
Continue. No worries.
Yep. Sorry about that. So to better visualize this, in case you couldn't hear it, there was 2 songs in each video, but on the left, you can see this is a max projection, and you see activity in this case. And on the right, you see the absence of activity. So even the bird is performing a very similar song, there's an absence of activity in the directed condition that we don't see in the undirected condition. Just to illustrate this a little bit more, here are an example 15 regions of interest, so 15 neurons during undirected singing from that video. And then here is the same 50 neurons just a couple of minutes later when the bird is singing to a female. And you can see there's largely a large absence of activity. And if I show you the summary data for this, you can really see on the left, this is behavioral, this is pitch variability for syllables for all the image birds. There's a reduction in variability, and that's really accompanied by a suppression of neural activity within area X. So to summarize this first part, area X SN activity was highly variable during undirected song and very active, and you could see it, and that was largely suppressed during directed song. And having this in mind, this was a really interesting contrast for us to see, and we wanted to ask next, well, if the low variability state is associated with less activity, could we suppress activity and get back a low variability state? For example, is area X activity necessary for vocal variability? And the way we tackled this was with inhibitory optogenetics, just to give us really fast reversible control over song. So we infected these neurons, these spiny neurons in area X with 2 strategies, either spiny neurons selectively or all the neurons in area X. And then we could implant a tapered optical fiber that allowed us to inhibit the activity of these neurons in a very quick manner, quick and reversible manner. And after validating this technique, we could then online detect the presence of a beginning syllable and trigger the laser randomly in, for example, a 30% of trials throughout the day. Then pool the laser condition and the not laser condition and just ask if the variability was different across the conditions. I'm going to skip a lot of quantification, just show you the upshot of these experiments, where for a single bird in the undirected condition, you could get a distribution that looks like this. In the directed condition, that's slightly compressed distribution. And in the undirected with laser condition, you get back something that I hope you can appreciate looks more similar to this directed distribution. It's tighter. And again, I'm skipping over a lot, but just to summarize this part, optogenetic suppression of variax activity was sufficient to reduce pitch variability to directed-like levels. Now, that was really interesting, and it prompted us to ask the next question, which was, if there is variable activity in this practice mode, this solo mode, how does it map onto vocal output? So while we're not also talking about learning today, we really hypothesized that this map existed, that information about vocal output was encoded in these patterns so that they can be used as a substrate for example, reinforcement-based learning of song during practice. That's part of what motivated us to look for this. This was easier said than done. It's not a trivial problem to address. Neural activity and vocal output are both very complicated signals with many dimensions. For example, you could have upwards of 100 neurons in one field of view, and you could have upwards of thousands of acoustic features that describe a spectrogram. So to address this problem, we collaborated with Jack Goffinet and John Pearson at Duke. And Jack and John had developed analytical techniques that would better help us address this question. So I'm just going to go a little bit into depth into what those techniques were. The approach.
So you have about maximum 2 minutes if we want to have any time for questions.
Okay, I might have to go 1 minute over in live questions, but I'll just try to hurry a little bit through this. The approach Jack took to address this problem was to first reduce the dimensionality of the acoustic and neural data into fewer, more informative dimensions, latent dimensions, that using a variational autoencoder approach that he developed in the past. And these latent representations still captured enough information so as to enable original reconstructions of the data. And a second advantage was that we didn't have to rely on hand-picked features or experimenter-defined features. And a new, unique thing that Jack added on top of this was a global latent that could allow him to model the joint distribution of the neural and vocal data and allow the two representations to conform to one another during the training. And having all this in place, our goal of the model was twofold. First, we could ask if we could predict neural and vocal latents from each other, and then we could also use the generative aspect to explore the relationship between neural and vocal activity. Now, in the interest of time and the fact that I only have about a minute left, I'm going to skip through the model performance and show you how we can explore the relationships between the data. Jack used canonical correlation analysis to identify an axis and understand whether neural and vocal data could be significantly correlated where projected through this axis. And for us, it was really exciting to see that these axes that could be found in the correlations were still significant on held out data, suggesting that the model had really identified an important relationship between SN activity and spectral output. And then we could use these correlation analyses, these correlation axes, to explore the relationship between the two data sets. We could randomly generate vocal neural data pairs of activity and project it along this significant correlation axis. And then we can ask for the spectrogram, what spectral information is more likely to happen on one end of the axis versus the other. So what you're seeing here is these contrast plots that highlight the spectral information that's more likely to be observed on one end of the axis versus the other, as well as the ensemble of neurons that are associated with those spectral variants. And this was really another strength of this approach. And so really having this in mind, we're now following it up in lab with further analyses and further experiments to explore these relationships using these joint encoding models. And I'll just summarize saying that these changes in SN ensemble patterns were correlated with highly structured changes in vocal output. And with that, I'd like to say thanks. And I'd like to just put a quick plug for Jack and John, who had really spearheaded a lot of this work. And they have an open joint position for anyone that's looking to be at the intersection between these new analytical techniques and neural techniques in songbirds, for example.
Thank you so much. So we have time for one question, or maybe more than one. Zahra Qasem Mohammed has the highest voted question, and they're asking, why do you think the activity starts before the onset of the song? Is this some sort of modulation from motor cortex?
Why the activity? Yeah, you probably saw that it starts before the onset of the song. Where you saw the dashed line, it was actually aligned to the first component of the core motif, but birds also perform little vocalizations in preparation, like little breathing inspirations right before. And we think a lot of that activity is also related to the preparatory inspirations that the bird performs before going into its main phrase. So bottom line, we think it's premotor activity as well that's linked to other small motor, vocal gestures that the bird is performing before. And we also, in general, tested whether auditory stimuli would elicit activity in these neurons, and we did not find that to be the case. Neither while the bird is singing nor offline playing back sounds of the bird's own song have no effect on these neurons.
The next question is by Kenji Doya. They ask, amazing. Is the suppression in DS by neuromodulation in area X, or lack of input from HBC or LMAN?
This is an excellent question. I didn't have time. I already was tight for time, embarrassingly, in this, but that is my whole second half of the project. It was exactly about this question. How does it happen we do not find it to be necessarily from HVC or inherited from HVC. Instead, we find that it's an adrenaline-dependent mechanism that can directly suppress these guys, these SNs. We tested this in slice. We tested this in vivo. We tested this at the behavioral level with microdialysis. And if you want more information about that, please feel free to just message me after.
Maybe one last question.
Sure, I'm trying to go fast.
Gautam Agarwal is asking, how much additional explanatory power does the nonlinearity of the autoencoder buy you? What if you were to use CCA between the spectrogram and neural firing? Short answer, please. And Preeya, please get ready.
Yes, that's a great question. I didn't have time to show you this again. In this talk specifically, we compared to a bunch of alternative models. One of them was a nonlinear version of the joint encoding model, sorry, a linear version of the joint encoding model. And we observed that the joint encoding model, it gave us a significant bump in prediction in R squared on the Tessa data between as compared to the linear version of the joint encoding model. And again, I can go more into depth into that offline, but the answer is it provided a significant boost in performance, the nonlinearities.
Thank you so much, Jonathan. In spite of the technical difficulties, you did a fantastic job. I'm glad.
Sorry about that.
So you can now mute yourself and turn off your camera and unshare. Make sure not to close, but unshare first. Oh, no. Okay. Hopefully it will work out. Preeya, can you turn on your camera and your microphone? And can you share, please, your slides just to make sure? Let's hope that everything works out.
Cool. Can you hear me?
Yes. If you have any slides that have audio, you might need to share with audio. Okay, perfect. So I'm very excited to listen to Preeya's talk, everyone. Preeya Khanna is a postdoc at UCSF, and she will tell us a little bit about how stimulation can help improve movement after stroke.
Great. Thank you for the introduction. So hi, everyone. Thanks for tuning in, and thanks to the Cosyne organizers for giving me this opportunity to present. So my name is Preeya, and I'm a postdoc at UCSF. And today I'm going to be talking about how to design stimulation to improve motor function in primates after stroke. So in the brain, networks of neurons in the motor system coordinate to drive movement. And after the most common types of ischemic strokes, pictured here with this X, some or most of this network is disrupted and can no longer contribute to the production of movement. And so this results in movement impairments, such as weak, variable, and slow movements. And unfortunately, in around 50% of patients, even after rehabilitation, they're still left with these movement impairments in their hand. So brain stimulation is a therapy that's promising for restoring hand function, since it can directly modulate the neural patterns in these underactive networks. However, how exactly to design the stimulation in order to improve neural and behavioral function remains unknown. So in this work, we're going to use a stimulation approach that our lab has found to be promising for improving movements in post-stroke rodents and test its efficacy in non-human primates while recording brain activity to answer this question of how stimulation might interact with neural activity during behavior. So our approach was based on a study from the Ganguly lab, which I'm in rodents, which used epidural direct current stimulation delivered through these skull screw electrodes in order to improve movement function following stroke. And so based on these results, we aim to translate this approach to non-human primate stroke model and to ask how stimulation interacted with these behaviorally relevant neural activity patterns. So we trained primates to perform one of two reach to grasp behaviors. After they plateaued in behavior, they underwent a stroke induction procedure that targeted primary motor cortex's arm and hand area. In the same procedure, we also implanted an electrode array in dorsal premotor cortex, or what I'll refer to in this talk as paralegional cortex, or PLC. And then we also placed skull screws, which were used for epidural stimulation later. So following the procedure, animals recovered, and we monitored their recovery on the reach to grasp behavior. And so on sessions where animals still exhibited behavioral deficits, we delivered stimulation, open loop 3 Hertz alternating current stimulation. So in this case, open loop refers to the lack of particular alignment of the stimulation waveform to the behavior that the animals were doing. So we just turned on the stimulation without particular alignment to the trial activity. And we chose to use a 3 Hertz sinusoid waveform as a compromise between the efficacious direct current stimulation approach that worked in the rodents and a desire to move towards a more clinically translatable charge balanced waveform. And so now I'm going to show you a video, there's no sound to this, where of one of our animals post-stroke. And so you'll see this animal performing the reach to grasp behavior, and what their objective is to be able to retrieve a pellet that's embedded in this well here. And so you'll see some of the movement deficits that I referred to earlier. And so you can see some of the difficulty that was associated with extracting this pellet. The finger movements were kind of slow and variable, and it was hard for the animal to manipulate the pellet as well. And so on that same session, we were able to apply alternating current stimulation and see how that affected performance. And so that was illustrated here. And so you can see that with the ACS, the animal was able to retrieve the pellet much more quickly. And so the answer to our first question was yes, we did find an effective epidural stimulation. And although I've only shown you 2 videos in one subject, we did test this approach in a total of 4 animals, each performing one of 2 reach to grasp behaviors, and found a strong effective ACS on grasping and hand function. And so this was really exciting and suggested that our approach might be clinically viable. But we also wanted to better understand the mechanisms of this stimulation and more broadly study principles of how the stimulation interacted with the neural activity. So there's a rich body of work studying how external fields interact with neural activity, both in slice, in vitro, and in anesthetized animals in vivo. And so common findings are that neural activity can be entrained to time-varying external waveforms, as illustrated here, and can exhibit changes in excitability. However, in the few studies monitoring neural activity in awake and behaving animals, it's unclear how movement-related patterns are modulated by these external fields. First, we don't know whether neural activity continues to be entrained to the external field, and second, if it does, how this entrainment and movement-related pattern interact. So in our preparation, we were able to first analyze the perilegional cortex activity we recorded during stimulation sessions. So this is an example of one of the units we recorded from PLC. This is its waveform and its inner spike interval distribution. And this is the probability of that neuron firing as a function of ACS phase. And so you can see that this unit strongly prefers to fire around the trough of the ACS phase. And so this is for one neuron, but we can also look across the full population. So we find that this neuron isn't alone, and that actually many of the units that we recorded in PLC prefer to fire around the trough. And so we defined this range of phases in which most of the units exhibited their preferred firing as the excitable zone. And so that referred to periods where ACS was able to increase excitability in the perilegional cortex. So having found these phases of excitability, we wondered how they might influence ongoing neural activity patterns and behavior. So one possibility is that it might be easier for movement-related activity patterns to modulate when trials start aligned to that excitable zone compared to trials that start outside of the excitable zone. And further, this might have implications for the success of behavior. So we first assessed the effect of trials starting inside the excitable zone versus outside the excitable zone on behavior. So we analyzed first reaching behavior, and we found that there was no significant difference between trials starting inside versus outside the excitable zone on reach duration itself. But then we analyzed grasping behavior, and we found that there was a significant improvement in trials that started inside of the excitable zone compared to trials that started outside. So given that trial alignment to the stimulation waveform influenced behavior and specifically influenced grasping, we next asked how it influenced the neural activity patterns. So this is an example of a trial average pattern from one animal in one session. The x-axis indicates time aligned to reach start, and the y-axis indicates units that have been sorted by their time of peak firing. And since we saw that alignment to the excitable zone had a strong influence on grasping but not reaching behavior, we wanted to split up this trial-related behavior into the reaching and grasping components too. And so we designated 3 different ensembles. So there's the reach ensemble, the reach grasp ensemble, and the grasp ensemble that you see pictured here. So when we analyzed trial-related activity, our main finding was using a metric called ensemble co-firing. And so that was defined as the fraction of total ensemble variance captured by the dominant shared dimension in the population. And so you can visualize this by looking at these pairwise correlation matrices. So this is for an example animal looking at reach grasp units. And so you can see that when you use data from trials that are outside the excitable zone, many of these pairwise correlation coefficients are lower than when you use data from inside the excitable zone here. And so this trend held up across the sessions that we analyzed. And so for the reach grasp and grasp ensembles, we saw significant increases in ensemble co-firing for trials starting inside the excitable zone compared to outside of the excitable zone. And so what we think is going on is a mix of two possible phenomena. So the first is that the alignment of the trial start to the excitable zone may create a slightly more synchronous burst of reach-related activity that may enable enhanced propagation of that burst to the reach grasp and grasp ensembles. And so indeed, prior work in feed-forward SynFire chain networks, which are just feed-forward excitatory networks, has shown that very small changes in initial burst synchrony can have consequences for downstream propagation. So these are two examples of activity propagating through a very simple SynFire chain network. Both receive the same input of 100 spikes, but at the top, that input is more diffuse, and here it's slightly more synchronous. And so you can see that in the top network, you get activity that becomes far more diffuse by the end of the propagation, whereas in the bottom, you have a nice synchronous activity at the end. And so what we think might be happening is that the alignment of trials to the excitable zone may enable a slightly more synchronous burst of reach-related activity that then propagates to reach grasp and grasp ensemble activity more synchronously. The second possibility is that ACS may itself have a direct influence on the neural population activity in the reach grasp and grasp ensembles. And so the increased co-firing that we might be seeing here might just be because when reach start falls within the excitable zone, you start to get grasp start can also fall within the excitable zone, depending on the exact timing of that particular trial. And so this increased co-firing may then improve propagation of grasp-related activity through later parts of the network and even possibly to downstream targets from the PLC that may account for the improved behavior that we see. So overall, we were able to find that ACS created these excitable zones associated with faster behavior and increased grasp ensemble co-firing. But we wanted to more clearly link the increased grasp co-firing that we saw to these ideas of neural propagation. And since co-firing and activity propagations have been well studied in SynFire chain network models, we adapted one of those to our behavior. So this is a schematic of the network that we used. So this is a SynFire feed-forward chain. It's composed of excitatory units that have solely excitatory feed-forward connections. Individual units are integrate and fire neurons. And the connectivity of this network makes it so that an input packet delivered here gives rise to a propagation of an activity cascade. And so within this network, we designated the first half of the network as a reach module and the second half as a grasp module, and analyzed it as though activity propagation was a proxy for successful behavior. So for the animal to successfully do a reach to grasp behavior, activity would have had to propagate all the way through the network. We then lesioned the model to recapitulate the stroke-related damage we saw in the experimental data. And by doing that, and we did that by pruning connectivity within the grasp module. And so you can see that for the same input, you get impaired propagation of activity through the network. We also added ACS. So in the experiment, ACS was delivered through a current injection on the skull screw electrodes. And in the model, ACS was modeled as an injected current into each integrate and fire neuron separately. So we were then able to analyze activity from the network and find the excitable zone. And we found that alignment to the excitable zone had consequences for propagation. So for an activity pattern that did not propagate fully, if we added ACS and had the input aligned outside of the excitable zone, that did not affect propagation strongly. But if we did align the input to the excitable zone, we saw consequences of improved propagation. We also saw increased ensemble co-firing for activity patterns that were inside the excitable zone. And in the network, we were able to directly link activity patterns that exhibited increased co-firing to activity patterns with better propagation activity. So here, for network patterns that had greater grasp co-firing, they exhibited also further propagation. And so what that said to us is that perhaps in our data, when we saw evidence of increased grasp ensemble co-firing, that reflected a network activity pattern that would also propagate further to complete the grasp activity pattern and also possibly to propagate further to downstream networks. So finally, we were able to also use the network to optimize stimulation parameters that we could test in future experiments. So one parameter we tested was the effect of varying stimulation amplitude. So we were able to see that if we had stimulation amplitude that was medium, we got good propagation. Increasing stimulation amplitude caused a truncation of endogenous activity patterns and actually inadvertently triggered some activity patterns. And very high amplitude totally swamped out the intrinsic network dynamics. And so in neuromodulation studies, often entrainment is seen as a good thing and evidence of good neuromodulation. But we noted that we could overdrive the system with ACS as well. We were also able to analyze the effect of frequency. And so we saw that low frequencies were efficacious and high frequencies were not. And we could even design waveforms that took advantage of these two principles. And so we were able to find that using this optimized waveform that combined the good part of the low frequency or the excitable zone, we could create waveforms that actually helped with propagation even when activity was not aligned to the excitable zone. So here with typical ACS, there was no improvement in propagation because the activity was not in the excitable zone. But with this new waveform, we were able to see better propagation. So overall, returning back to our first question of how can we design stimulation to improve motor performance, we found that ACS improved grasping. We found that ACS created these zones of excitability, and that alignment to the zones of excitability improved both grasping behavior and co-firing compared to not aligning to those zones. And finally, we were able to use the network model to link together our observations of grasp co-firing to better activity propagation properties. And so with that, I'd like to thank the Ganguly Lab and my advisor, Dr. Karunesh Ganguly, for creating a great environment for doing this work, our collaborators at USD, and all the folks at the California National Primate Center who contributed to this work. And I'll just say that our paper that kind of described these results just came out last week, so feel free to check that out for more details or also get in touch. With that, I'd be happy to take any questions.
Thank you, Preeya. Beautiful work and congrats on the paper. We look forward to looking at it. There are a number of questions. Before that, I'll maybe ask very quickly since we have time. Does the effect sustain after the stimulation? Like if you do it repeatedly, does it actually improve behavior when you're not being stimulated? And if so, how long does that take?
Yeah, that's a great question. So we actually found no effect of sustenance of the behavioral improvements. So on some sessions, we did do stimulation where we started the day with no stimulation, then stimulated, then did a sham block after that. And we didn't really see any persistent effects. And we think largely that's because this approach is kind of directly modulating the neural activity. But yeah, it's really very intriguing. We imagine that if the approach is able to improve behavior, that you could imagine the brain's intrinsic plasticity mechanisms being able to consolidate that and give rise to recovery later. But we don't have the experiments to support that right now.
Thank you. So the first question is from Nicholas. They ask, does the effect of stimulation on behavior start on the very first session of stimulation, or does it take time for behavior to improve with stimulation?
Yeah, that's a good question. So we didn't find any evidence of kind of the animal having to adapt to the stimulation. So if we just look at trials, we do see kind of immediate effects kind of as soon as stimulation turns on in the behavioral session. But I will say that these animals are recovering from a stroke, so we don't have thousands of trials which we might need in order to assess an effect like this. But in our hands, we don't see kind of an adaptation to the stimulation.
So Yang Fan asks, as you use a SynFire model, do you assume that the local cortical network is feed-forward? Do you have indications in your co-firing for feed-forward activity?
Yeah, so we chose the SynFire chain model because of its history in kind of studying how synchrony affects propagation. And we thought that given that we saw co-firing in our data, that's why we kind of made the selection of this model. We also selected a model kind of that had simple connectivity so that we could understand the activity pattern really well and move forward with designing these optimized stimulation waveforms. But we're not claiming that the brain has these SynFire chains kind of embedded in them. We know that there are, inhibitory neurons, definitely more complicated connectivity like recurrence. And so this was just a model to allow us to kind of move forward and understand the implications of how stimulation can interact with simple network dynamics. But yeah, I think it would be really interesting to add complexity to the model and test some of these new stimulation waveforms in a more complicated model before testing them experimentally.
A related question comes from Guillaume Lajoie. He asks, is there an explicit feature of the stroke model in the SynFire model, and how did stimulation affect it?
Yeah, so this will be, yeah, so what we did in order to model the effect of the stroke was we pruned connectivity within this GRASP module. And so what that gave rise to was these activity patterns that had kind of good propagation through the reach module and poor propagation through the GRASP module. And so in stroke, we know that certainly a lesion kind of gives rise to very damaged tissue, but it also gives rise to kind of increased inhibition and lower firing rates in general, since that damaged tissue also has connectivity to other intact areas. And so lesion, kind of pruning connectivity was our approach to damaging this SynFire chain network model.
Kenji Doya asks, how does ACS cause intracellular current injection?
Yeah, so in the experiment, we delivered ACS through these electrodes, and that gave rise to what we think are, electric fields that kind of span the cortical surface. And so this was just a first pass way of modeling that effect on individual cells. We think certainly you could add complexity to the model by modeling these voltage gradients and then modeling the effects of the voltage gradients on the cells themselves. And then you can start to even incorporate more complex features too, like geometries of cells and kind of how they align to the voltage field. But since we saw evidence of neural entrainment in our data, using a current injection was a very simple way of recapitulating that feature in the model. And then since our interest was in how does entrainment kind of give rise to changes in behavior-related patterns, this was our first pass way. But yeah, in the brain, we think that the cells are really responding to these voltage fields, not a direct current injection.
And before we go to the poster session, maybe one last question from Sebi Rolotti. They ask, they say, great talk. Do spontaneous movements tend to be initiated at a specific phase of intrinsic rhythms around 3 Hertz?
Yeah, so we did look for, at least in our experiment, evidence of the animal preferring to initiate movements in the excitable zone versus outside of that. And we didn't see evidence of that from our data. And again, these are in kind of impaired animals that aren't performing so many behaviors. But yeah, in kind of animals performing spontaneous behaviors or in healthy animals, we do see that the frequency of those movements tends to be around 1 to 3 Hertz. So we do see sub-movements that fall within that frequency range. And so, yeah, we think that this frequency is well matched to kind of the behavioral frequency that we might expect to see both in healthy animals and maybe as well in stroke animals.
Thank you so much for efficiently answering so many questions. And of course, Preeya can answer further questions in a private sessions. And also, I think that the questions from Slido will go to you. See you all at the poster session. Thank you, Preeya.